The cell cycle consists of three parts that allow for the division of cells. The longest part is interphase, which accounts for about 90% of the cycle. Interphase is comprised of three subphases: G1 phase, S phase, and G2 phase. Cell growth occurs during G1 phase. Chromosomes are copied during S phase and more growth occurs, and G2 is where the cell completes its growth and prepares to divide. The mitotic phase of the cell, M phase, includes both mitosis and cytokinesis. Mitosis is the division of the nucleus. Cytokinesis is the division of the cytoplasm and usually immediately follows mitosis. The mitotic phase of the cell, M phase, includes both mitosis and cytokinesis. Mitosis is the division of the nucleus. Cytokinesis is the division of the cytoplasm and usually immediately follows mitosis. Mitosis is essential to all living things. It allows organisms to grow, develop, replace dead cells, repair damage, and allows some single-celled organisms to reproduce asexually. Mitosis consists of four phases: prophase, metaphase, anaphase, and telophase. During prophase, the chromatin fiber condenses to form discrete chromosomes, which consist of two sister chromatids joined at their centromere. The nuclear envelope begins to break down. The mitotic spindle begins to form. The nucleoli disappear. Centrosomes move away from each other, and each chromosome has a kinetochore. In metaphase, the centrosomes are at opposite poles of the cell. The kinetochores of the sister chromatids are attached to the kinetochore microtubules coming from opposite poles, and the chromosomes are aligned on the metaphase plate. Metaphase is the longest stage of mitosis. Anaphase is the shortest stage. The sister chromosomes are separated and brought to opposite sides of the cell as the kinetochore microtubules shorten. The non-kinetochore microtubules lengthen, causing the cell to elongate. During telophase, the nuclear envelope is reassembled and the nucleoli reappear. The chromosomes become less condensed. Mitosis is complete with the formation of two genetically identical nuclei, each containing the diploid number of chromosomes. Cytokinesis causes two daughter cells to appear shortly after the end of mitosis. In animal cells, a cleavage furrow forms and pinches the cell in two. In plant cells, a cell plate forms. Meiosis allows for the production of gametes, which allow for sexual reproduction and variation. Meiosis is divided into two parts: meiosis I and meiosis II. Meiosis I separates homologous chromosomes, and meiosis II separates sister chromatids. Each part has a prophase, metaphase, anaphase, and telophase, and cytokinesis. During prophase I, the chromosomes begin to condense, and homologous chromosomes begin to loosely pair up. Crossing over occurs, providing an additional source of variation. The nuclear envelope breaks down, the spindle forms, and chromosomes move towards opposite ends of the cell. The microtubules from the poles attach to the two kinetochores. The homologous pairs move towards the metaphase plate. During metaphase I, the homologous pairs line up on the metaphase plate so each one in a pair is facing a pole. The chromatids of the homologs are attached to kinetochore microtubules from opposite poles. In anaphase I, the homologs separate and are moved by the spindle apparatus towards opposite poles. Telophase I usually occurs simultaneously with cytokinesis. A cleavage furrow occurs in animal cells, with the cell plate forming in animal cells. The result is the formation of two non-identical haploid cells. Each chromosome is composed of two sister chromatids. During prophase II, the spindle apparatus forms, and the chromosomes move towards the metaphase II plate. In metaphase II, the chromosomes are positioned on the metaphase plate the same way as in mitosis. The kinetochores of the sister chromatids are attached to microtubules from opposite poles. The sister chromatids are not identical because of the crossing over that previously occurred. Anaphase II consists of the separation of the sister chromatids and their movement to opposite poles. During telophase II and cytokinesis, the nuclei form and the chromosomes become less condensed. The result of meiosis is four genetically different haploid cells. Mitosis and meiosis each have functions that are essential to all living things, though each play different roles. Both involve the replication of DNA through interphase. Mitosis consists of one division, resulting in two genetically different diploid cells. Meiosis consists of two cell divisions, resulting in four genetically different haploid cells. The cells produced in mitosis are identical to the parent cell, while the ones produced in meiosis are not. Crossing over occurs only in meiosis. Both mitosis and meiosis consist of prophase, metaphase, anaphase, telophase, and